Hello, dear students. In this video, we will be learning about cellular respiration. By the end of this lesson, you should be able to do 4 key things. First, you should be able to identify the major structures of the mitochondrion. Second, you should be able to correctly sequence the stages of cellular respiration. Third, you should be able to calculate how much ATP is produced from one glucose molecule. Finally, you should be able to compare ATP production in aerobic versus anaerobic respiration. Keep these learning targets in mind as we move through the lesson. They will guide everything we discuss. This essential question connects today's lesson to broader ideas in biology. How do cells extract energy from food molecules? And why do they use multiple metabolic pathways? Food provides cells with chemical energy stored in molecules such as glucose. Cells cannot use this energy directly, so they convert it into ATP or adenosine triphosphate through cellular respiration. During cellular respiration, glucose is broken down in controlled steps. The energy released is captured in ATP, which serves as the cell's immediate energy source. When ATP breaks down into ADP or adenosine diphosphate, it releases energy that powers cellular activities. Cellular respiration is the set of metabolic reactions that cells use to convert energy stored in nutrients into ATP or adenosine triphosphate. The overall chemical formula for cellular respiration summarizes how cells extract energy from food. C6, H12O6 plus 6O2 react to form ATP plus 6 CO2 plus 6 H2O plus heat. Glucose is the fuel molecule. Its chemical bonds contain stored energy. Oxygen is needed to help break these bonds efficiently. As glucose is broken down during cellular respiration, carbon atoms are released as carbon dioxide, and hydrogen atoms combine with oxygen to form water. The most important product of this process is ATP, which cells use to perform work such as movement, transport, and building molecules. Heat is also released as a byproduct, contributing to body temperature in animals. The key idea here is energy transformation. Chemical energy stored in glucose becomes usable cellular energy in the form of ATP, which cells use to perform work. Breathing is the physical process of taking in oxygen and releasing carbon dioxide. Cellular respiration is the chemical process happening inside cells that uses oxygen to make ATP. The diagram explains how oxygen enters the body through breathing, travels in the blood, and enters cells. Inside the cells, oxygen becomes part of water during cellular respiration. At the same time, carbon dioxide is produced and carried back to the lungs to be exhaled. Breathing supplies oxygen for cellular respiration and removes carbon dioxide produced by it. ATP powers every activity in your body, including heartbeat, breathing, nerve signals, and brain activity. In fact, the brain alone uses about 1/5 of the body's total energy. The slide also introduces basal metabolic rate, or BMR. This is the energy your body needs just to stay alive, even when you are resting. Cellular respiration constantly supplies ATP to meet these energy demands. The mitochondrion is a specialized organelle found in all eukaryotic cells. Its main function is to produce ATP, the cell's usable energy, through a process called oxidative metabolism. Mitochondria are located in the cytoplasm of nearly all eukaryotic cells. Cells that need more energy usually contain more mitochondria. Structurally, the mitochondrion has 2 membranes. The outer membrane is smooth, while the inner membrane is highly folded. These folds, called cristae, increase surface area and allow more energy-producing reactions to occur. Because of these membranes, the mitochondrion has 2 compartments: the matrix, which is inside the inner membrane, and the intermembrane space, which lies between the 2 membranes. Proteins located on and within the inner membrane carry out oxidative metabolism. These proteins use oxygen to break down macromolecules and generate ATP, supplying energy for cellular activities. This diagram shows the endosymbiotic theory. An early host cell engulfed an aerobic bacterium that could use oxygen to produce energy. Instead of being digested, the bacterium lived inside the host cell and provided large amounts of ATP. Over time, this bacterium evolved into the mitochondrion. This evolutionary event occurred before plants and animals diverged from a common eukaryotic ancestor. As a result, all eukaryotic cells, including both plant and animal cells, inherited mitochondria. Although plants have chloroplasts to make glucose through photosynthesis, they still need mitochondria to break down that glucose and produce ATP. Animals, which do not photosynthesize, also rely on mitochondria to produce ATP from food. This is why both plants and animals contain mitochondria today. This diagram shows the 4 stages of aerobic respiration, the process cells use to release energy from glucose when oxygen is available. Let us walk through each stage briefly. The first stage is glycolysis. Glycolysis occurs in the cytosol, outside the mitochondrion. In this stage, one glucose molecule is broken down into 2 smaller molecules called pyruvate. A small amount of ATP is produced, and energy is also captured in electron carriers. Glycolysis does not require oxygen, but it is the starting point of aerobic respiration. The second stage is pyruvate oxidation. This stage takes place in the mitochondrion. Pyruvate molecules are converted into acetyl coenzyme A. During this process, carbon dioxide is released, and more energy is transferred to electron carriers. This stage prepares molecules for the next cycle. The third stage is the citric acid cycle. The citric acid cycle occurs in the mitochondrial matrix. Here, acetyl coenzyme A is completely broken down into carbon dioxide. A small amount of ATP is produced, but most of the energy is stored in electron carriers that move on to the final stage. The fourth stage is oxidative phosphorylation. This stage takes place along the inner mitochondrial membrane. Electrons carried by NADH and FADH2 move through the electron transport chain. Oxygen is required at the end of this chain. This stage produces the majority of ATP made during aerobic respiration. Together, these four stages allow cells to efficiently convert energy stored in glucose into ATP that powers cellular activities. Let us investigate further the four stages of aerobic respiration. Glycolysis takes place in the cytoplasm of the cell. During this stage, one glucose molecule is broken down into two molecules of pyruvate. This process releases a small amount of energy. The products are 2-pyruvate, 2-ATP, and 2-NADH, or nicotinamide adenine dinucleotide. Glycolysis does not require oxygen, but it prepares molecules for the oxygen-dependent stages that follow. The second stage is pyruvate oxidation. Pyruvate oxidation occurs in the mitochondrial matrix. The 2-pyruvate molecules produced during glycolysis enter the mitochondrion. The 2-pyruvate molecules are converted into acetyl coenzyme A. During this conversion, carbon dioxide is released, and energy is transferred to NAD+ to form NADH. No ATP is produced at this stage, but acetyl coenzyme A is formed to fuel the next step. The third stage is the citric acid cycle, also known as the Krebs cycle. This stage also occurs in the mitochondrial matrix. Acetyl coenzyme A enters the cycle and is completely broken down. Acetyl coenzyme A is broken down into carbon dioxide. The products include 2-ATP, 6-NADH, 2-FADH2, or flavin adenine dinucleotide, and carbon dioxide. Most of the energy released during this stage is stored in electron carriers rather than ATP. On the left side of the slide, you see a photograph of Sir Hans Adolf Krebs, the scientist who discovered this cycle. He identified how cells convert food and oxygen into usable energy, and received the Nobel Prize in 1953 for this work. In his honor, the citric acid cycle is often called the Krebs cycle. On the right side of the slide is a circular diagram, which shows how the citric acid cycle functions as a repeating pathway. The circular shape is important because it emphasizes that the cycle continues as long as acetyl coenzyme A is available. The fourth and final stage is oxidative phosphorylation. This stage takes place along the inner mitochondrial membrane. NADH and FADH2 donate their high-energy electrons to the electron transport chain. Oxygen acts as the final electron acceptor, forming water. The energy released as electrons move through the chain is used to produce about 28 ATP. Because this stage produces the majority of ATP, it is the most energy-productive stage of respiration. This slide summarizes how much ATP is produced at each stage of aerobic respiration and where that ATP comes from. On the left side of the slide, we see a list breaking down ATP production, while the diagram on the right shows where each stage occurs within the cell. Overall, this slide emphasizes that most ATP is produced during oxidative phosphorylation, while earlier stages generate smaller amounts and focus on transferring energy through electron carriers. When oxygen is not available, some cells produce ATP through anaerobic respiration, specifically fermentation. Glycolysis still occurs, producing 2 ATP per glucose molecule. Fermentation then converts pyruvate into ethanol and carbon dioxide, while NADH is converted back into NAD+. This regeneration allows glycolysis to continue. Fermentation does not produce additional ATP, but enables cells to survive short-term oxygen shortages. When oxygen is not available, some cells use lactic acid fermentation to continue producing ATP. Glycolysis still occurs, producing 2 ATP per glucose molecule. Pyruvate is then converted into lactate, while NADH is converted back into NAD+. This regeneration allows glycolysis to continue. Lactic acid fermentation occurs in some bacteria and in human muscle cells during intense exercise, when oxygen supply cannot meet energy demand. To answer this lesson's essential question, cells break down glucose and other molecules through aerobic and anaerobic respirations to efficiently release stored energy as ATP. They use multiple pathways to adapt to different oxygen levels and maximize energy extraction under varying conditions. To review, Define the main function of the mitochondrion, locate where glycosis takes place, and count how many ATPs are produced in aerobic respiration. Thank you for listening.